hydrogens gives on this side. So today hand we are dealing with a topic called as structure of proteins. Called as structure of proteins. So let's get started. Now, here every polypeptide chain is synthesized by the polymerization of amino acid monomers in a specific sequence, which means it's a long chain and you have a lot of different types of amino acids here, and ultimately it will be a long chain. Then only it will be called as a polypeptide chain. And here the polypeptide chains which are forming the protein are in accordance with the genetic information which is encoded in the molecule of the mRNA, so called as a blueprint of the life, which brings the information transcribed from a gene. Okay, deal with. Now, the polymerization process of amino acids take place on the ribosome, and hence it is called as translation, or what we call as a protein synthesis. Okay, deal with. Here, each type of polypeptide chain undergoes bending and folding in a specific pattern to assume its three-dimensional structure. Which means if I am drawing a single line, this is not just like the protein structure. Rather, this will be something a protein structure, and for making this structure, it has to go through the secondary structure, then tertiary structure, or maybe it will be reaching up to the quaternary as well. Okay, deal with. So here, only then, like it will be dealing with its three-dimensional structure. Only then the protein will become capable of its specific function. Okay, deal with. Here, the pattern of the bending and the folding is genetically inherent. The pattern of bending and folding of the protein chain is genetically inherent in the amino acid sequences. Okay, deal with. Now, there are a lot of different types of the structure of the protein. For example, you will be dealing with the primary structure, then the secondary structure, tertiary structure, and at last but not the least one called as a quaternary structure of protein. Okay, so let's get started now. Here, starting the first thing first, called as a primary structure of a protein. Here, okay, deal with. So here, the specific linear sequence of peptide bonded amino acids which are making a polypeptide chain are representing the primary structure of a protein. So basically, the bond which is present in two amino acids is called as a peptide bond. The bond present in the two amino acids is called as a peptide bond. I'll just draw here. Okay, so this is just like an amino acid for me. Here I am drawing the NH2 group, which means the amino group. Here I am drawing the COOH, which means the carboxyl group. Here I will be drawing the H, that is the hydrogen, and here I will be drawing the R1, that is the alkyl group for me. The second amino acid here will be coming, it will be having the first amino group, I say NH2, then the carbon, then the carboxylic. Here it will be having the R2, that is the alkyl group second, and here will be having hydrogen. And both of them are forming the peptide bond, so the new structure will be CONH. Then here C, you had here H, you had here R2, and the COOH. Correct? And here you will be having, your chiral carbon, here R1, here you are having your hydrogen, and here you are having your NH2 group, which means the proper bonding pattern has been taken place here. CONH, or what we call it as peptide bond. It is a covalent bond and the most important characteristic of a protein. Okay, so whenever the word peptide is coming in front of you, do remember that the first amino acid is reacting with its carboxyl end, whereas the last amino acid is reacting with its amino end. Which means in a protein chain, the first amino acid always have a free N terminal, whereas the last amino acid always have the free C terminal or the carboxyl terminal. Hopefully you are getting the case. Now, here methionine and cysteine are two different amino acids which have sulfur in their structure. We have already discussed this in the yesterday lecture. that the methionine and the cysteine have sulfur in their R group, or what we call as alkyl group. Okay, deal with. Now, in case of the cysteine, we have sulfhydryl group. We have sulfhydryl group, which means SH, okay, deal with. Now, basically, here if I'll be going for, we may have the disulfide bonds as well. Like specifically in the cysteine only, because there are two types of the cysteine. Like the cysteine are joined to make a cysteine molecule. Okay, deal with. We'll be discussing that as well. Now, dealing with the secondary structure here. The secondary structure of the protein. So here in the primary we only had a single line which has amino acids attached in due to with the help of only single peptide bonds only. But still that is not a complete structure. So we have to deal with the secondary structure as well. So here in case of the secondary structure, the polypeptide chain becomes folded into two types of secondary structure. The polypeptide chains become folded into two types of structure, that may be the alpha helical or the beta pleated sheets. Okay, deal with. And the special bond which is formed in the secondary structure is the hydrogen bond. The special bond which is formed in the secondary structure is called as a hydrogen bond. Please do remember the case that in case of the primary structure it is the peptide bond, whereas in the secondary structure it is the hydrogen bond. It is formed in between the oxygen. It is formed in between the oxygen of the carbonyl group and the hydrogen of the amino group. Oxygen of the carbonyl group and the hydrogen of the amino group. Deal with. Now, in a helical structure, one or more segment of a polypeptide chain becomes coiled in spiral formation. So ultimately you will be observing this type of structure. Okay, deal with. Now, here because throughout the length of a such segment, the carbonyl oxygen of each amino acid becomes hydrogen bonded to the amino group of the fourth amino acid along the chain. Do remember one thing, that first amino acid is bounded first, second, third and the fourth. And if these two are getting bounded with the help of a hydrogen bond, now the structure will be something like this. Correct? First, second, third, fourth, and here you have a hydrogen bond. So ultimately like the structure will become this. And what we call it as helical structure. Deal with. Now, here the segments of a polypeptide chain come to lie side by side, and they are held together by the special bond called as hydrogen bond between the carbonyl and the amino group of the first and the fourth amino acid. Okay, deal with. Here, because here like ultimately we are dealing that in case of the alpha carbon which is attached with the functional groups directly, the alpha carbon which is forming the backbone of a polypeptide chain, to which the alkyl group chain of each amino acid is attached. They are successively located at a little above or little below to the plane of axis. And that is the only reason it will be forming a zigzag like structure. Okay, deal with. Now, and this zigzag like structure will be called as a pleated sheet, or we will be calling it as beta pleated sheet. So basically majority of the secondary structure proteins are in the form of the alpha helix, but some of the proteins are in the form of the beta pleated sheet as well. Okay, deal with. Now, in case of the multimeric proteins, which means a lot of different subunits are there. So there maybe two or more polypeptide chains are linked together. Maybe there are two or more polypeptide chains which are linked together, and they are also forming due to the interaction between the segments of the chains. So here a polypeptide chain may acquire only a helical or only a beta sheet. Or the combination of both, which means the alpha as well and the beta as well. Please do remember one thing, that this is beta. Okay, deal with. Now, here You can observe here the structure as well, like the alpha helix and the beta pleated sheets as well. So this is none other than the alpha helix. This is none other than the alpha helix. We have literally gone for the structure as well. But I'll tell you one thing, that the example of the beta pleated sheets, because like that is one of the most important question in case of the competitive exams. So the examples for the beta pleated sheets are, the first example is a silk fibroin. The first example is a silk fibroin, and the second example is a wool. Yes, wool, sheep wool. So do remember one thing, that the beta pleated structure of the protein is observed either in the silk or in the wool. Either in the silk or in the wool. So you can literally deal with. And one more example here they have given, that the hairpin motif of the beta sheet. Hairpin motif of the beta sheet. So you can observe that here like you are having a slightly curve, whereas here you are having straight chains. That is the reason that the alpha helix is something like this and the beta pleat is something like this. Okay, hopefully you are getting the case. Now here I'm dealing with a slight detail of what the super secondary structure is. So ultimately here in case of the super secondary structure, it will be the combination of both the helix as well and the beta pleat as well. The helix as well and the beta pleat as well. Okay, deal with. Now dealing with the tertiary structure of the protein, I'm dealing with the tertiary structure of the protein now, and for that case, most proteins of cells, majority of the proteins of the cells which are occurring as compact, and they are roughly spherical or globular, because in the formation of the tertiary structure of the protein, you will be going for formation of the any type of the bond. They don't need to go for like only the peptide bonds should be formed or only the hydrogen bond should be formed. No. In case of the tertiary structure of the protein, you can form any possible bond. You can form any possible bond to reduce the surface area of the protein. To reduce the surface area of the functional protein. Do remember. So here, to attain the tertiary level of a protein structure, a polypeptide chain is undergoing further folding to pack its secondary structure to make it further close in the structural formation. So here you will be having in the formation of this domain, the R group side chain of the distant amino acid residue come close together to play their roles in the protein function. So ultimately now you will be forming any possible bond. Here you can observe as well. Here you can observe as well like this kind of structure. You can literally observe that they have gone for supercoiling. They have gone for supercoiling. So you can literally conclude in case of the tertiary structure of the protein, any possible bond can be formed. Okay. Here we will be writing here. Whereas the folding of the polypeptide chain into the secondary structure depends only on the hydrogen bond between the carbonyl and the amino group. Here in case of the tertiary, you will be having the 5 different types of the interaction, which means all the interaction. 5 different types of the interaction between the R group side chain of the amino acid residues, which means here like the alkyl groups will be coming into picture. The alkyl groups will be coming into the action and they will be forming any type of the possible bond they can, so as to minimize the surface area of the protein. The first one is none other than the disulfide bond. We have already discussed about in case of the cysteine. We have already discussed in case of the cysteine. So do remember. The another one is called as electrostatic, or what we call as the ionic bond, which means the sharing the lone pair of the electrons, specifically the charged atoms will be forming this. Even the hydrogen bond you can form. Yes, you can form the hydrogen bond in between the carbonyl and the amino as well. And whatever the molecules are, whatever the atoms are present there, they can share the hydrogen bond. Even you have the weak bonds as well called as the van der Waals forces, and you can have hydrophilic or hydrophobic interaction as well, which means this combinedly I can call any possible bond should be formed to minimize the functional proteins surface area. Okay. So like the question will be arising here in our mind that if the tertiary structure of the protein is forming any possible bond, what is the role of the quaternary structure then? There is no type of the bond as left. So here in case of the quaternary structure, whereas the secondary and the tertiary protein structures are characteristics of single polypeptide chains, the quaternary protein structures are occurring only in the multimeric proteins, which means it may have 2 or even more. And how many? Up to dozens or even hundreds of the polypeptides of tertiary level, which means two tertiary proteins can make a quaternary protein, but maybe there will be hundreds of tertiary proteins are coming together and making one single quaternary protein. Okay, deal with. So here, the most cellular proteins are enzymes. The most cellular proteins are enzymes, and most enzymes and other proteins are quaternary in structure. And the example I should give you is the insulin and the hemoglobin. It's the insulin and the hemoglobin. I hopefully like you do know, but still insulin is made up of two different polypeptide. One of them is 21 amino acid long, and the other is 30 amino acid long. And if you are joining them, it will become 51 amino acids long chain. So insulin has how many amino acid? It is as 51 amino acid. The other case is about the hemoglobin. So hemoglobin is a literally a tertiary, like ultimately here. This is hemoglobin made up of alpha, alpha dash, beta, and beta dash. Okay, deal with four different chains are here, and they are the globular proteins of the quaternary structure. They are the globular proteins of quaternary structure, or rather I should say they are one of the abundant proteins in the animal body. The component polypeptides, which means the multimeric subunits in the quaternary proteins, are held together by the same interactions which are responsible for the tertiary structure in the single polypeptide. Which are responsible for the tertiary structure in a single polypeptide. So basically, once a protein is folded in its native conformation, in its proper conformation, it can also associate non-covalently with other proteins as well. Non-covalently with other proteins as well, which means if a protein is stable now, if a protein is settled now this are like this target protein can literally associate with another proteins as well in a non-covalent fashion. In a non-covalent fashion. So you can deal with that case as well. Here you can have like the another as well, like the not even with the proteins, you can have the bonding with the nucleic acid as well, you can form with the lipids as well. That is what we call as the like the lipoproteins as well, or the proteolipids as well, to form the supramolecular complexes. Here in case of the viruses, multifunctional enzyme complexes are present. Here in case of the viruses, the multifunctional hopefully this lecture has been beneficial for you guys. Thank you very much for your so much cooperation. Thank you. Again, I'm asking you, please do comment on our lectures, because whenever you are commenting on a lecture, those literally those lectures are getting much more higher ratings as compared to those which have not been commented by you. Okay? Like, share and subscribe.